We are now going to be recording the rest of the lecture, and this will then get put on the web. Did anybody try to download the PowerPoint with audio? Did that work for you? Okay. The PowerPoint with audio file is rather big, and I hope that the one that they're doing now should be smaller than that. That is and has in the past. In addition, depending on people's systems, Icon did or didn't work, so I usually try to mount it both at Icon and on Google. So we'll see if we can work it that way as well. I will be emailing information about how to find all of these as we go along. In general, when there's a new chapter, we'll have a new handout here. We'll try to have the previous handout, if they're still available, to this side. Don't take 2 copies unless you've managed to use up completely the previous one. And try to bring it with you to class, because typically handouts are good for at least 2 lectures, sometimes even for 3, and I'll try to point out when that's true. Are there any questions about the things we discussed on Wednesday, or about course organization in general? Questions? All right, well, let's plow ahead then. We finished up by talking about polysaccharides as the last topic on Wednesday. And so I'd like to move down to the next bit of the topic. In addition to these building blocks, which we're going to see, and we're going to see both how they're very flexible in terms of what the body can do with them, and very tied to their function, that is, the structure of the molecule will always reflect its function. We're also going to need to think at least a little bit about energy, because energy is what drives metabolism. Metabolism is, in broad terms, the transformation of matter and energy, being able to capture it, being able to do something different with it, use it for a new purpose. That's only possible when there's some free energy available to drive those reactions. And so chemists for over a century and a half have tried to think about the energy content of reactions and how that can go forward. And one of the more interesting dimensions of energy that is useful to think about in biochemistry is the Gibbs free energy, which is symbolized by the letter G. The Gibbs free energy is the sum of the enthalpy, which is a fancy Greek way of saying the heat content of a system. And the heat content is not the same thing as temperature. You both all know that if you have a piece of hot metal and a piece of hot cardboard, you can touch the cardboard but not the metal. The metal has a higher heat content. And entropy as a function of temperature. Now, entropy is a little more subtle. It's not quite as easy to conceive of as heat content. Entropy is a way of thinking about disorder. And this is one of the classical representations of an increase in entropy. Here you have your pool table before the game starts. Here you have it after someone's broken it. There's only one way to lie up the balls in a triangle. There's not unlimited, but a very large number of ways to lay up the balls like this. And so this is said to be in a state of greater entropy, greater disorder. The absolute Gibbs free energy is difficult to measure. So what we tend to work with is changes in free energy. The delta here is the Greek capital D. Deltas are often used for differences. In this case, we're talking about differences in free energy, differences specifically in Gibbs free energy, and that can be calculated or related to the change in heat content and a change in the entropy at a given temperature. The important thing to remember is that a reaction will be spontaneous if a Gibbs free energy change is less than 0. If the Gibbs free energy change is less than 0, if the products have less free energy in them than the starting stuff, the reaction will probably be spontaneous. You can go spontaneously from this to that. You have to put energy in to get back from that to that. Questions about that? This is things some of you have encountered at least twice before here at the university, but experience tells me that it has to be put into a context for it to be sticking. Now, life would come to an end if every single reaction had to have a negative delta G. And fortunately for the world and for us, you can couple a spontaneous reaction to a non-spontaneous one and make that one then go forward. And just to give sort of a arbitrary example of that, you can have one reaction where A goes to B, a second where B goes to C. If the reaction A goes to B has a negative free energy change here, minus 20 kilojoules per mole, and the reaction from B to C has a positive free energy change, a positive Gibbs free energy change associated, the overall reaction, A to B and then B to C, or A to C, has the sum of those free energies. So you can add the free energies. And since the sum of them is a negative free energy change, this reaction is in effect pushing the other reaction forward. If it came downstream of it, you could think of it as pulling it. But you can couple a spontaneous reaction to a non-spontaneous one, and that's what makes all of metabolism work. Sort of a major overview of this concept and as how it applies to metabolism is in this schematic of what might be called the core of metabolism. All of life starts from carbon dioxide in the atmosphere. Carbon dioxide is a fully oxidized carbon. Going from an oxidized carbon to a partially reduced carbon, like in a monosaccharide, would not be a favorable reaction under normal circumstances. But if we put in light energy through photosynthesis, we can do that. Now this has potential energy associated with it. When it becomes oxidized back to carbon dioxide, it can release free energy for whatever purposes you want to apply it to. So oxidation and reduction are often features of metabolism, and by cleverly coupling them, we can make it possible to make the whole scheme go in the direction we want it to go in. I pointed out to you a previous table that I wanted you to be sure you were refreshed on. That was table, this figure. That was slide 9, table 1.1. If the textbooks have not arrived by the time that I get back to my office, I will put that table on the web in a larger, more magnified version. And same will go for this. Among the various things that you should notice about functional groups is their oxidation and reduction states, because that is a sign, a visible sign of whether they are, in effect, higher energy or lower energy forms of a compound. Here you have carbon dioxide at the top of this. All 4 bonds of that carbon are to oxygen. That is a fully oxidized carbon. It is the least reduced carbon. And you can have different reduction states of carbon all the way from this fully oxidized form down to methane, which is the fully reduced form. It has hydrogens on all of its bonds. It's the least oxidized. So a bond in which the carbon is sharing with an oxygen is generally one where the carbon has been oxidized. A bond where the carbon is sharing with a hydrogen is generally a case where the carbon has been reduced. And the more reduced it is, the more energy there is. We have methane gas coming out of our stoves. We don't have carbon dioxide coming out of our stoves. And this series of energies reflects the electronegativities, the fact that oxygen can steal electrons from carbon, and carbon can steal electrons from hydrogen. But if you look in between here, you're going to see many intermediate states of oxidation and reduction. You can see double bonds. You can see keto groups here. You can see aldehydes. I don't think they have there's an alcohol here, there's an alcohol, and so on. So these are groups that we're going to see again along the way. Have you run out of handouts up there? Do we need more up there? Yeah. Okay. Lynn, could you take some more of the new handouts up there? Thank you. Okay. It really is important to try to leave that territory empty, guys, because this is a big campus, and as long as I've taught this class, there are people who come from a distance where they can't get here by 3:30. So it is a kindness to your fellow students. Okay. So how did all of this get started? Where did the complicated machinations of biochemistry get started from? And this is a question that's been of interest to people for a very long time. And it's been handled in a biochemical way for a few decades, since about the Second World War. A clever experiment was done of trying a sample in which a mixture of gases that was considered to be a mimic of the early atmosphere of the Earth was energized by electrodes in it, creating, in effect, lightning bolts, shooting lightning into it. And this mixture of gases was run around through boiling water or simmering water so that you could have it recollect, have aqueous reactions happen, and so forth. And certainly in the samples that were removed, a number of interesting compounds were observed that are considered organic molecules. A drawback to this experiment is that it used an atmosphere of gases in here that was more reducing than we now think the early atmosphere of the Earth was. On the side of favoring the idea that molecules can form spontaneously is that the variety of molecules that were formed in this setup overlap to a considerable extent with the organic molecules that are observed to form spontaneously in interstellar space and that are observed on meteorites that land on the Earth. So there is definitely a certain capacity for carbon atoms to become more complicated just in the course of things, just because they bang around the universe and they run into nitrogens and oxygens and hydrogens. So carbon has an innate tendency to get complicated. When we look at Earth itself, some of the interesting places to think about this happening, one of the more recent sort of favorite spots, are places called the hydrothermal vents. And if you look at an underwater map of the oceans, you're going to see places where the plates are colliding, and there tend to be areas where hot water is streaming up that's been heated by this kind of special geological location. And those hydrothermal vents provide a number of features that have caught people's attention in terms of contributing to making a rich pre-biological soup. They include some heat, so there's a source of energy, if you like. These vents often have reduced compounds in them, and as I pointed out, most biological synthetic things involve a certain amount of reduction to make things more complicated. The hydrothermal vents have colder water right nearby, so you have a place where things can be cooled off and kept nice. And in the stuff that comes out of these hydrothermal vents, we do see even today spontaneously forming things like acetic acid, short polypeptides, just spontaneously. Another sort of candidate to be suspicious of are clays. Clays are a group of minerals that share the features of having positive charges on their surface. And the proposal has been made that those repeating positive charges allowed for the alignment of monomers to facilitate the formation of a polymer, because the clay served as the catalyst, if you like, for making a more complicated molecule. Another feature of the hydrothermal vents that makes them an interesting target for investigation, if not necessarily the guilty party, is they are still occupied by organisms that are very peculiar and very old. That is, we can deduce the age of organisms by doing geological trees, and some of the life forms that are present in those vents can be deduced on that basis to be a very old form of organization indeed. I'm not going to go into a lot of discussion of it right now, but I want to throw into the sort of pot the latest suggestion on possible places where origins of life might have happened. And this is called soup in a sandwich. That one problem in thinking about the hydrothermal vents is the water's washing past it, and you might wonder, how could you accumulate enough for something to happen? And the soup in a sandwich hypothesis is talking about the fact that mica is a mineral that naturally forms very thin sheets that has positive charges on their surfaces, like the clays, is rich in potassium, like the interior of cells, and provides a lot of little niches, little spaces in which these reactions could happen and not get washed away. So this is definitely an area of ongoing reflection and hypothesis forming, and I think things will continue to surface. I'm not quite sure what happened to that slide. I'm sorry. One of the things that has to happen for life to form is there has to be replication. There has to be some way to make more copies of the thing or make more copies of the information. And what this diagram is showing you is just showing you what replication really means. This isn't itself a mechanism because they're not showing you anything that's helping you make it happen. But this is what replication means. If you have something to begin with, that you need some way to align with it, either the same or a complementary form of that thing, to then have those polymers separate, and now you can bring in new partners and repeat that process until you have copies of both partners. There is actually quite an interesting recent finding, which is that a non-biological aromatic hydrocarbon can act as a midwife, they like to say, to this replication process. This is a heterocyclic ring that can fit between bases, and this molecule can simply help to pack and help polymers form in the absence of any enzymes. So there is now a non-biological assistant that could facilitate replication, something that we did not have even a glimmer of until the last few years. Some of the proposals for early life suggest that this polymer would have been an RNA-based life, where RNA was both the gene and the enzyme, and we may talk about that more later. One of the difficulties with that theory is that RNA tends in our world to be pretty unstable. I don't know if any of you have worked with RNA, but if you kind of sneer at it, it falls apart. But again, quite recently, in the last few years, were discoveries that borates are a mineral that stabilize RNA by a very simple chemical mechanism. And so here again is something that's not biological, that's available in the environment that can facilitate these potentially simpler forms of doing things. Questions? Okay. Let's take a little break for the diagnostic test. Does everybody have a copy of the diagnostic test? Does anybody need one? Questions? Could you guys help out with getting some of these around? Thanks. This is, again, for your use. Take a moment to keep your hand up if you need a copy, please. Okay, I'll get over here. Right. There you go. All right. This is just meant to give you a chance to see whether or not you have the background that I'm assuming you have. Other people, who needs it in this row? The diagnostic? Just that one? Okay, pass that down, please. Anybody else need the diagnostic? Up there, okay. There you go. Do you need some more? Sorry. Thank you. I don't have a copy now. let's see if I can pull this off. Pop that down. Pop over to here. And is this going to open? All right. Okay. Do folks need any more time, or have you pretty much a little more time? Okay. You can talk to each other if you have any questions on what you're uncertain about. This isn't going to be graded. This is for your own use. So if you have any questions you're not sure about, please do talk to your companion, your next door neighbor. So. And for the benefit of any students who are listening to this on the web, I will just mention I will post the answers as well. So for question 1, what would be the correct answer? D? Okay. And in question 2? Okay. Question 3? C? B. Okay, there we go. How about D? What? I mean, sorry, 4. What about 4? Yeah, it's Friday for me, too. Question 4. Sorry? Okay, there we go. F? I mean, 5? Sorry, I'm human. Thank you. 6. 7. I'm not quite sure what I was hearing, but 8. D, right? Okay. 9. Yeah. And as many dogmas, there are now exceptions to this, but we'll get to that. And 10? Okay. All right. And again, this is not a very strenuous exam, but if there were more than one question you had any difficulty with, be aware of the fact that you might want to brush up or just pay extra attention going along. Okay. So let's go back then to where we were talking about cells. Folks? Okay. These are times. I want you to pick 1 or 2 blocks that you want to work on. Okay. The bottom row is after class and evening. So, okay. So one of the things that happened when cells got started was that they acquired a really unusual capacity. And we're going to see how unusual when we get to photosynthesis, which is the ability to fix carbon, to take carbon dioxide out of the atmosphere and turn it into organic molecules. That's clearly going to take a lot of energy input because it's a very oxidized atom. Almost as soon as carbon fixation happened, photosynthesis also happened. And photosynthesis is a brilliant invention because it allows cells to capture sunlight and use the energy of sunlight to do the carbon fixation and to do the other kinds of work that have to happen for metabolism to work. So whereas before photosynthesis happened, it was probably driven off of the chemical energy of rocks, once photosynthesis was involved, most of the rest of life became dependent on solar energy. Very soon after the first forms of photosynthesis, a brilliant form of photosynthesis became available. Oops, sorry, let me back up a page. I keep forgetting that clicking here means something different. I think I know why. What happened was that the blue-green algae came up with an ability to use water as the donor for the electrons, and in the process, they released oxygen. They didn't mean to release oxygen. It was just a waste product as far as the blue-green algae was concerned. But the oxygen that the blue-green algae released became the basis for the rest of life, or the rest of eukaryotic life. All of these activities, fixing carbon, photosynthesis, and generating oxygen, were going on as early as 3.5 billion years ago. So these things have been shaping or reshaping the face of the Earth for a very long time. We see traces of blue-green algae or cyanobacteria, to give them their other name, as far back as 3.5 billion years. So these oldest forms of cells are what we call prokaryotes, before nuclei. And within the prokaryotes, we distinguish between two groups that are biochemically rather different from each other. Eubacteria, and you probably recognize the word bacteria. Eu here is just the Greek for good or true. And then archaea, which comes from the Greek word for first. And these were named because they thought they were the very first forms of life. That may not be true, but that's the name they were given and were stuck with. What I'm going to say about prokaryotes is a bit different from what you'll see in the textbook. And so this is as good a place as any to introduce my policy in this class, which is when I in the textbook disagree, I'm right until you show me a published article showing me I'm wrong. So it's worthwhile to pay attention in lecture. I will try to point out the places where I'm correcting the textbook. But if I've corrected something in the textbook, you can't then say on the test, but the textbook said, in this class, I'm a higher authority than the textbook until you show me a published peer-reviewed article. There aren't very many places where the textbook needs correcting. I just want to mention that there are a few. In prokaryotes, there are either single or no internal membranes. That is, all cells have a membrane around them. There has to be a membrane around them for them to be a cell. And in the prokaryotes, there are either no internal membranes at all, or there are single membranes, just things that make a plain old vacuole or a flattened vacuole or something like that. But prokaryotes are definitely capable of having internal membranes. In general, prokaryotes are going to have some kind of a wall. There are some naked prokaryotes, but they mostly have some kind of a wall. They store their genetic information in circular DNA, usually. One of the other things you'll find in biochemistry, if you haven't already from your biology class, is there are very few 100% statements in biology. But in prokaryotes, almost always have circular DNA. And that circular DNA is packed into what's called a nucleoid. It's kind of like a nucleus. And you can see the nucleoids over here as dense areas within the cells. So these are areas that have a lot of DNA and protein and RNA packed tight, and so they're very dense. Prokaryotes are usually small, less than a micron, but there are definitely exceptions to that, and we'll point out some of those. One of the hallmarks of the prokaryotes is they have complex biochemistry. The strategy that prokaryotes take to adapt to weirdness in life is to develop new biochemistry. And so prokaryotes can do things like live off of gold ores. They can live in boiling water. They can grow on rocks. One of my more favorite ones was when they wanted to come up with an enzyme that would degrade testosterone. The way they did that was they looked at the dirt around fire hydrants and isolated the bacteria that grow in that soil because it's an environment that's testosterone rich. And so they were able to find bacteria that had enzymes that break down testosterone. They're very, very clever in their biochemistry. Prokaryotes, because of their compactness, and their density and their relative simplicity in structure show the greatest extremes of where they can live. They can live in boiling water. They can live in sulfuric acid. They can live in the Dead Sea. So high salt, high pH, low pH, high temperature, low temperature, high osmotic conditions. Basically, any physical variable you can think of, you can probably find a prokaryote that can tolerate it or even flourish under it. And this is actually a very useful thing. If any of you have used Taq polymerase stands for Thermophilus aquaticus, and it is a hot springs organism that was isolated from basically boiling water, and that's why its enzyme will work again after it's been raised up to 90 degrees centigrade, and so on. We're very used to thinking about the unicellular bacteria, but not all prokaryotes are unicellular. There are definitely prokaryotes that make multicellular masses or hyphae or whatever you want to call them. And there are definitely prokaryotes that are capable of having several cell types, more than 2 cell types in the case of some prokaryotes. And in some cases, these are terminally differentiated cells that will never do anything else again. So the developmental complexity of the prokaryotes does extend to the point of being multicellular a bit in some species, definitely can have more than 1 cell type, and some of them can make terminal differentiation. Any questions about any of those before we move on? My perspectives on prokaryotes are shaped by the fact that I knew the blue-green algae or cyanobacterium well before I knew any of the others. And you tend to have a lot of respect for prokaryotes if you meet them that way. To give you a little bit of a sense of how big or small these different things are, here are some typical bacterial cells. These have all been saved on a filter surface. We're looking at them by a scanning electron microscope now. Well, these are scanning microscope. This is something else. And this gives you sort of a ruler, if you like, a typical bacterial-sized cell. Here are some virus particles. Each little green dot there is a virus. So you can see how much smaller a virus is than a typical bacterium, or typical virus than a typical bacterium. And here's a red blood cell. The red blood cells are not very big cells as eukaryotic cells go. They don't have a nucleus, but you can see that they're a good deal larger than the bacterial cells. So about the eukaryotes, how are the eukaryotes different from the prokaryotes? Well, all eukaryotes have internal membranes. They have a variety of internal organelles that are bounded by membranes. And some of the organelles in eukaryotes have double membranes, where they have one complete one and a second complete one around that one. And the two that are most important to keep in mind would be the mitochondria, which are the powerhouses, the source of most of the ATP for cells, and the chloroplasts, where photosynthesis goes on in eukaryotes. Not in prokaryotes. Prokaryotes do their photosynthesis differently. The defining mark of a eukaryote is going to be a nucleus. There are eukaryotes that don't have chloroplasts. We don't have chloroplasts. There are eukaryotes that don't have mitochondria. We can show by their genes that they are parasites that have lost them, but they're still eukaryotes in other respects. But all eukaryotes are going to have a nucleus, and that nucleus will have a double membrane around it, and that double membrane will have very specialized pores to permit things to go in and out. Inside of that nucleus, there is no protein synthesis. The nucleus in eukaryotes is a specialized domain for the processing of information. It's a place where you have chromosomes of DNA, linear chromosomes in virtually all cases, that are going to generate either more DNA, so you can have more cells, or they're going to generate RNA that then provides for making proteins and so on. So there's a domain for that kind of metabolism, and then the cytoplasmic domain for the rest of metabolism. Eukaryotes may or may not have a wall. If they have a wall, that wall will usually be cellulose, which is not generally seen in the prokaryotes. If one speaks statistically, usually eukaryotes have a wall because most eukaryotes are plants. But that's strange to think about. Eukaryotes come in a very, very wide range of sizes, a much larger range of sizes than the prokaryotes do. Eukaryotes have complicated morphology. Where prokaryotes can come up with really weird biochemistry to do things, eukaryotes can come up with really weird morphology to do things. And so we're very adept at having 5 different kinds of chewing teeth and fingers versus fins versus wings versus, all kinds of stuff. So the morphology of eukaryotes is far more complex than the potential range is for prokaryotes. Eukaryotes pay for this complex morphology with limits in space. The eukaryotes cannot get into such strong acid. They can't get to such high temperatures. They can't live in such high salt. So the eukaryotes are a bit more fragile than the prokaryotes. Eukaryotes can become very complicated, but they're not as robust as the prokaryotes. Any questions? In this picture, what I've tried to do here is to show you, here we have one of those bacteria from the previous slide that was the sense of scale. So that's the bacterium. And then this is a euglena, which is a single-celled organism that's capable of living on its own. So it performs all the functions of the organism. And you can see the abundance of intracellular organelles here. And some of those organelles are basically the same general size as the prokaryotic cell. So it's almost like this eukaryotic cell is a small city, or at least a small village of prokaryotes in terms of bringing things together, not necessarily their chemistry. Other questions? I want to emphasize that it's not an all-or-nothing thing, that there's a general scale to these things, but that range can be quite broad. And so if we look at prokaryotic cells, most prokaryotic cells are going to be, a bit less than a micron to maybe up to 10 microns. But there are prokaryotic cells as big as 100 microns. There are a few prokaryotic cells that are a full millimeter across. You can see things with your eye when they're a millimeter. So there are prokaryotic cells, relatives of bacteria, you can see with your naked eye. In the case of eukaryotic cells, they tend to be in that range of, 5 to 100 microns. That's sort of the size that most people have in their heads when they think about eukaryotic cells. Some eukaryotic cells get quite small. Some eukaryotic cells are actually smaller than a micron. And we're going to see something startling about that. But the upper end gets quite big, so it's not that unusual to find eukaryotic cells that are more than a millimeter across. And there are eukaryotic cells that get up to, here's a meter, all right? We're about this tall, in about there. There are eukaryotic cells that are bigger than us. And you might think, goodness, where are those? Think about a whale and the way neurons work. Whale neurons are several times longer than you are tall. So eukaryotic cells can be very, very long indeed. And this wide range of scale at the cellular level is also shown to some extent at the level of the multicelled eukaryotes. So once you get away from the protozoans who are a single cell, multicelled eukaryotes tend to be generally in the range of about, this would be here, would be this would be about a millimeter here, so about 10 millimeters or a centimeter here. Multicelled eukaryotes between a centimeter and a meter. That's a very common range to find multicelled eukaryotes in. There are some kind of small ones in there. And mammals have a smaller range themselves. To give you some examples to go along with that sense of scale, these are some of the ones to fill in here. There is a prokaryote called Prochlorococcus that is only 0.6 microns. You don't need to copy them down because they are in your handout. Prochlorococcus is barely half a micron. It's just a little more than half a micron. And it is one of the most abundant photosynthetic organisms in the world. It's a classic example of you get what you fish for. When oceanographers started out, they went fishing with nets that were, 25 microns across. And everything they got was bigger than 25 microns. And then they started using 10-micron nets. And everything they got was bigger than 10 microns. And it wasn't until they started using submicron filters to look at things that they could find the cells that were less than a micron across. So the Prochlorococcus is one of the most abundant photosynthesizers, only discovered in the last small period of time. Cyanobacteria quite routinely get a lot bigger than bacteria in general, the blue-green algae. Cyanobacteria are quite often 100 microns long, 50 microns across. They can be quite big cells, quite commonly. And there is, again, relatively recently discovered a prokaryote called Thiomargarita that is a millimeter across. It's a sulfur bacteria, and it looks like a little pearl, a little millimeter pearl, but definitely a prokaryote. If we look at eukaryotic cells, a larger range of size than the prokaryotes, there's a group of algae called the picoalgae, the tiny algae. And Ostriococcus is one of these eukaryotic cells that is less than a micron across. And it, again, is one of the most abundant eukaryotic photosynthesizers. So the atmosphere that we breathe, a significant chunk of it is coming from organisms that are less than a micron across. Then we want to look at some of the larger things. Within bodies, oocytes and nerves are large cells, but the ciliates and the forams are actual organisms that can be 100 microns, can be a millimeter, can be a centimeter across. Individual cells that you can see, that you can actually pick up if you have good fingertip control. And then the nerves that can get quite big. And then I've gone ahead and given you some examples here of both extremes of these things, of how there are eukaryotes that are actual multicellular organisms that are less than a centimeter, and ones that are kilometers across. So the mammals tend to be more compact in their scale. Any questions? Okay. These are just some pictures of them. And you can see here that Ostriococcus, the smallest eukaryote, is actually smaller than the blue-green alga that's going through division. This will also show you how many internal membranes this prokaryote has. Looking at the interiors, the interior of the blue-green algae isn't obviously simpler than the eukaryote, but the eukaryote has a nucleus. And that's, here's the nucleus. The eukaryote has a nucleus, and that makes it definitively a eukaryote. We can deduce the evolution of cells from a couple of sources of kinds of information. There are biochemical clues, how they do their metabolism, what kind of molecules are present. And there are also morphological clues of what kinds of structures are there and how are they related. When we look at the eukaryotes, which would include animals, fungi, and plants, we have some characteristics from both archaea and bacteria. It's not a real... it's not a black or white case of what made it across the divide. If you find these questions of interest, I have a number of supplementary readings on the ICON website that I can recommend to you. If these are questions that interest you and you find this level of it very straightforward, I would encourage you to take advantage of those to deepen your knowledge of this. So where did the eukaryotes come from? One proposal has been that the chloroplasts are descended from the blue-green algae or the cyanobacteria, and that the mitochondria are descended from other kinds of bacteria. But there are a lot of compartments in the eukaryotes that don't have any genes. Chloroplasts and mitochondria have small genomes of their own that is part of the basis for making these relationships to the algae or the bacteria. Another difficulty with this theory about an anaerobic cell eating blue-green algae or eating a potential mitochondrion is, in the realm of cells that we know, anaerobic cells don't do things like eat other cells. Eating a cell is a very energy-demanding activity. And so if eukaryotes arose that way, it would be an unusual thing. An alternative way of thinking about it is that the initial large cytoplasm got compartmented and that there was specialization within the compartments. There's a proposal that was made for where the nucleus comes from, which is often not discussed, and I believe I have a reprint to this as well, that you started off with some prokaryotic cell with the DNA attached to the membrane, as it often is in prokaryotic cells, and that this membrane body that was growing around it that we see in prokaryotic cells, completed the enclosure of it, so it created a compartment around the genome, and just needed to develop passages between that to let things in and out into the cytoplasm. And I just want to sort of raise the question, if invagination and specialization could make a nucleus, why couldn't it make a mitochondrion? But I'm being a heretic here, so I just raise it as a possibility. What's left in the handout at this point are some supplementary things from the textbook to let you take a look at them and refresh your memories of them. Actually, if we take a look, we might see that a lot of them are here already. Yeah, so in your handout, are these large enough to see readily? Okay. So you do have a copy then of many of these groups here. So this is the content of table 1.1. So you do have it in your handout already at, I think, a large enough size to make out. And then, yeah, this is more of table 1.1. Okay, great. So I don't think we'll start on the water, chapter 2. Now we have about a minute or two left before the end of the period. Do people have any questions about the material we've talked about today or about the course in general? Have a good weekend, and I'll see you Monday.